Okay, ladies and gentlemen, so listen, the point of this particular lecture is just to sort of have a quick and dirty review of some of the basics that you got in chemistry last year, just to make sure that we're up on it for our biochemistry unit that we'll be doing, because you really want to make sure you have a good grasp of this. As always, take notes. If you have questions on specific things, bring them to the Zoom on Wednesday. Reach out to me during the office hours, email me, and let me know, and I will gladly go into more detail about anything that you might need. Okay? So, terms to know. Now, in your notes, you're going to type these in. You'll notice that I've given you a great deal more space, just because I thought it would be easier that way. So, we have element, which is a matter composed of atoms that have the same atomic number. My atom is the smallest component of an element that still has the properties of the element, right? We've got a positively charged nucleus, a negatively charged electron cloud surrounding it. Protons are positively charged component in the nucleus, and it has an atomic mass of 1. A neutron is non-charged, also has an atomic mass of 1. Electron is negatively charged and has a negligible mass. We don't really calculate it in when we're calculating atomic mass. And then an isotope is an atom with the same number of protons and electrons, but a different number of neutrons. Okay? So 95% of living matter is made up of these 4 different atoms: carbon, oxygen, hydrogen, nitrogen. Those are the big 4. Okay. We know that as humans, we are carbon-based life forms. Everything on this planet is a carbon-based life form. I will be giving you a link to the Hank Green crash course in biology, and it's about how carbon is a tramp. It's fantastic and does a really nice job going into more detail on some of these things. So, atomic structure, right? You have your nucleus, which contains your protons and your neutrons. In order to find the atomic mass of an element, you make sure that you add up the number of protons and neutrons. Okay? Protons, obviously positive charge, neutrons, no charge. My electron orbitals, my innermost shell holds only 2. The second and third shells each hold 8 electrons. Bluntly, in biology, at least what we're doing, we're not going up much past those 2 orbitals, okay? And an element's reactivity is determined by the number of electrons in the outermost shell. The closer it is to being complete, honestly, the more volatile it is. If you look at your one column of the periodic table where you have chlorine, okay, we use chlorine to kill stuff because it's volatile, okay? So that's, that stuff's pretty volatile. It'll sort of snag anything. Your stuff in the middle tends to be a little bit more on the sharing end of stuff. So electrons move around the nucleus in these atomic orbitals. Again, their chemical reactivity is determined by the electrons. Overall, your element wants to become stable. Mother Nature is big on this, okay? Everything in nature wants to become stable, whether it's grounding an electrical charge or an atom wanting to have a neutral charge, okay? The atom is most stable when the outermost energy levels has 8 electrons, and that stability can be achieved by either adding or losing electrons or even potentially sharing electrons. Your redox reactions we'll go into a little bit more detail on later. Transfer of electrons from one molecule, that molecule is oxidized, to another, which is reduced. I know I struggled with this for a really long time. What I finally wrapped my brain around is the one that is being reduced is, yes, I'm adding electrons to it, but I'm reducing its charge. That's why it is saying that it is being reduced, because I've actually now given it a negative charge. Okay, so that helped me once I finally wrapped my brain around that one, because I was always like, why is it reduced? I'm adding crap to it. Now that's why, because I'm reducing the charge of it. So I have some different kinds of bonds and forces of attractions. You have covalent, which is sharing electrons. We know that because co means together, right? Ionic is the transfer of electrons and making ions. And then I have hydrogen bonds, van der Waals forces, and hydrophobic interactions. Okay. So covalent bonds, this is where I'm actually sharing electrons. I have both polar and nonpolar covalent bonds. Listen, what you need to know is that nonpolar that's an equal sharing of electrons, whereas polar is unequal sharing of electrons. I am not going to ask you on a test or a quiz what exactly the electronegativity difference is or anything like that. But I do want you to know that the difference between polar and nonpolar in an electron sharing standpoint, okay? Ionic bonds, this forms, we have ions that are being formed because I'm either gaining or losing electrons. And then I now have two charged particles that are then attracted to one another. My negatively charged ion is referred to as an anion. I always knew that because I had the extra N in it as opposed to the cation that was positive. Okay, that has the T in it, that makes it a plus, and plus is positive. And this is also known as an electrostatic attraction. If you look over to the right there, sodium fluoride is sort of our classic example of this, okay? Chlorine gains an electron, sodium loses an electron, and then they're very happy together, and chlorine is no longer something that's going to kill us if we breathe it in because, you know, salt can be bad for you and too much, but, you know, it's pretty much an okay thing. Now, hydrogen bonding. This is bonding between molecules, and it's based on the attraction between a hydrogen on one molecule and either the nitrogen, oxygen, or phosphorus on another molecule. Of your just sort of molecular interactions and forces of attraction versus a bond, this is the strongest. It is not stronger than a covalent or an ionic bond, but it is stronger than hydrophobic interactions and van der Waals forces. Really important in stabilizing the 3D structure of proteins and nucleic acids. So the rungs of the DNA ladder in between the two backbones is actually a hydrogen bond. And we'll talk why that's so important later on when we get into DNA in specific. And it's involved in enzyme catalysis. So here's the thing. And we'll get into a lot of specificities later. We'll do a whole thing on enzymes, but right, I've got an enzyme, I bind it with my substrate, and then hydrogen bonding puts pressure on it to either form a bond or break a bond, okay? And this is really important for that. Van der Waals forces are attractions between electronically neutral molecules. It's a weak attraction. Little bit of heat destroys it. But again, this is helpful in my 3D structure proteins, and it also is important for the structure of the cellular membrane, okay? Now, water, okay? Water is really, really stinking important. And you're going to have to know the properties of water, hint, hint, wink, wink, nudge, nudge, for the quiz you're going to take on this, plus your biochemistry test that I'm going to be giving you, okay? Not that you have to have it memorized, but you need to understand the concepts of it. So living things are 70 to 90% water. It is a polar molecule. Because it is polar, that makes it the universal solvent. Now, like dissolves like, you should have learned that in chemistry last year, right? Polar substances, polar liquids dissolve polar solutes. But it is very important, and it does make it the universal solvent because of this. We have hydrogen bonding to one another. This strengthens the connection between molecules, which also gives it a much higher boiling point and a higher freezing point. Okay, so for example, alcohol both boils and freezes at a much lower temperature than water does. Okay, and it's really important. So our body is, I think humans, if I remember correctly, are something like 75% water. It might be even higher than that. I'm not remembering off the top of my head. Anyway, we don't want to get so hot that we internally begin to boil, okay? Freezing point is higher. This is important because that way living things, you have this whole ability to, you know, first of all, it doesn't freeze within our bloodstream, but it impacts like the environment and whatnot, and it keeps water as a liquid for longer. It facilitates important chemical reactions in and out of living organisms. We're going to talk about hydrolysis later on. They are cohesive, which means that they bond to one another. They are adhesive, which means they bond to other things. Okay, the example I've given here is the meniscus in a graduated cylinder, right? You have to read the bottom of it because the water sort of goes up the side a little bit. It has a high heat of vaporization, and Water is the only substance of which I am aware that is less dense as a solid than it is as a liquid. It's really important because this means the ice floats, which means that then things like frogs and fish can live underneath it all winter. Acid-base review. Okay, I'm not going to get into Lewis acids and bases. You're welcome, but we are going to talk some. So an acid is a compound that releases a large number of hydrogen ions into the water. That H plus is a hydrogen ion. My examples here are hydrochloric acid and hydrosulfuric acid. Okay. A base releases hydroxide, that's that OH negative ions into the solution, and this would be sodium and potassium hydroxide, which are the examples that are given here. Okay. pH measures the hydrogen ion concentration. Okay. Anything less than a 7 in the pH scale is an acid. Anything greater than 7 is a base. And a 7 itself is considered neutral. Now listen, organisms must maintain a relatively constant pH. Our bloodstream is somewhere around 7.4. If we deviate much from that, we die, which by the way is also all these people that are like, oh, if you have an alkaline diet, then it's the drug companies keeping cancer cures away from us. And if you have an alkaline diet and make your blood alkaline, then you'll never get cancer is a bunch of crap, because you can't make your blood alkaline because otherwise you'll die. Plus our body, you have to put a lot of alkalinity into your body in order for it to actually begin to do that to the point that it causes problems. So, you know what? Here's the other thing I want to go over. I'm sorry, I just thought of this. Here's something that people sometimes get confused on. The higher the pH, the lower the acidity. Okay, the lower the hydrogen ion concentration. Some people get really confused over that. They're like, oh, high acidity, high hydrogen ion concentration means high pH. No, the higher the hydrogen ion concentration is, the lower the pH is. I'm just throwing that out there because, like I said, it does confuse some people sometimes. pH indicators are a weak acid or a weak base, and they change color when exposed to certain hydrogen ion concentrations, and that change varies based on the indicator that is used. So, common indicators: litmus paper. My blue litmus paper turns red in the presence of an acid. My red litmus paper turns blue in the presence of a base. Phenolphthalein goes from colorless to pink in the presence of a base. I've always thought that's a pain. It would have been really nice if Mother Nature had just worked it out so that everything turned blue in the presence of the base, but nope, that's not the way it works. It's nice. Buffers are used to maintain a pH balance, so it either takes up or releases hydrogen ions. We have this in our bloodstream. It's called carbonic acid, and it is what helps maintain a consistent pH within our bloodstream. And that is that, ladies and gentlemen. So again, if you have any questions or concerns, please do not hesitate to ask.